everybody, and welcome to 17 Minutes of Science, the Quarantine Chronicles. I've been thinking that maybe we'll start calling this something else, like the Phase 1 Opening Chronicles. But in the meantime, we're still not moving around very much, so we're glad to have you joining us today. My name is Sarah Cheesman. I'm a technical solutions scientist at In Vivo Biosystems, and thanks for hanging out with us today to hear about a really interesting topic with a great title, Worming Our Way into Machine Learning, how we are using machine learning for variant classification in C. elegans. And our guest today is Dr. Lan Guo. So Lan is a data scientist at In Vivo Biosystems and is our machine learning expert. And she spends her days thinking about how to apply this form of artificial intelligence to modeling genetic variants, as well as understanding complex phenotypes and big sets of transcriptome data. So from time to time, I hear Lan say things like supervised learning and artificial neural networks, and I just think those are fascinating concepts and a whole different way to look at the world. So as we get going, feel free as always to drop us a line over Facebook, let us know you're there, ask questions, and otherwise raise your hand. So Lan, welcome back. This is our second conversation that we've had on Saturday. But if you could just give us another brief introduction about yourself.
Sure. Yeah. My name is Lan. I am a data scientist at In Vivo Biosystems. I have a background in neuroscience. I used to do research with neurophysiology, and then I switched into the data science world that I'm in right now. So now I spend my day digging through data that is still biological data that we generate through our human avatar C. elegans platform, and we're trying to study disease.
Well, thank you for qualifying how it all works, because I think for a lot of people, this is a pretty abstract concept. So conversation will be enlightening. And that leads me into the first question, which is in which ways does machine learning allow us to answer questions we wouldn't otherwise be able to do so if I guess it was just human brains on the problem?
Yeah, well, first human build machine learning, so it's still human.
I guess that's true.
Yeah, I think of machine learning as a good tool in that it's kind of, I think of it as machines or computers trying to find patterns in data and try to formulate it in sort of a parameterized mathematical model. And you can iterate over and over again and make a model that can be used for future decision making. So the difference to human, I would think, is more of the mathematical modeling framework, and the computer can take in and keep in memory a lot of data or parameters at a time, more than human can, and can iteratively do mathematical operations on these parameters to arrive at some kind of conclusion that is better than human or more precise than human can. So that's the advantage for computers there. Also, I think that human may sometimes have some biases or preconceived notion, like we would go in hypothesizing that this variable would affect that variable, but the computer would not have, well, if we don't encode that kind of bias into a machine learning model, then usually during our exploratory data analysis phase, we will be able to explore multiple different hypotheses. And sometimes we would discover interesting things that are unexpected.
Yeah, that's really interesting about the human bias. Now that you say that, that's quite obvious that a computer can be asked not to do that.
Yeah, we have to be aware of our own bias, and in that we don't want to bias our model. So it's a little bit meta. Yeah. And also, like machine learning, I mentioned like finding pattern in data. So maybe put a little context around that is, why do we want to find patterns in data? So for example, we might be interested in some kind of physiological data parameters that would predict the outcome of a disease, let's say. So that's why we're interested in finding variables that are important associated with the outcome of interest. And that's usually what supervised learning does. And that's what we do most in our job at In Vivo, is to try to find things that would predict genetic variants association with disease.
Mm.
Yeah. And if there's other type of machine learning, like unsupervised learning or reinforcement learning, that might just be interested in finding patterns that are carrying certain information or of certain properties. Yeah. But I hope that doesn't go too technical into the weeds.
No, it's that phrase again, supervised learning.
Yes, trying to echo back to you.
Yeah. So piggybacking on that, I mean, we're talking about using whole organisms, and that would be the worm system, C. elegans, as the experimental model. And so what are the most challenging things that you've encountered in trying to model C. elegans data using machine learning?
Yeah, I think that this is not specific to C. elegans, but maybe pretty common in biology is that the data is usually messy. So there's a whole bunch of stages that we do to do this whole, machine learning is not, it's one big concept, but it comprised of many different stages that of different type of work that has different challenges. So for example, we start with biological, physiological data. And that data is sometimes messy, so you need to actually spend a lot of time wrangling the data to massage it into a form that is, is high quality and valid before you could even start doing any modeling. It's because garbage in, garbage out. So you want to make sure you have things that carry the information of interest first. Mm-hmm. So there's a lot of data engineering, data pipeline design that go into controlling the data quality, cleaning up the data, how do you deal with missing data, all this good stuff. So the goal there, and then it goes into feature engineering. So picking out sometimes you don't want to just put raw data into a model. You want to first distill some higher level information from the data to help your machine learning model make a better prediction. And all the purpose of this is to get the data into a right representation to then put into your algorithm. And then once you get to that, you have to choose the right learning algorithm or framework or model, whatever you want to call it. There's so many out there. So you have to make a decision there. And then you do your prediction, your model, whatever, then you have an output. Then you need to be able to interpret it. You need to be able to tell how this model, which is a machine, not a human, how did it make the decision it did. You cannot just ask him, why did you think this one is pathogenic? Is it my answer? So you have to be able to interpret and interrogate the internals of this model to tell, to like make a decision or kind of carry some information for the human decision maker to say the doctor or the scientist interested in the hire us to do the work with, tell them why we arrive at this decision, right? So yeah, that's the interpretation part. And yeah, so there are many different stages and each one has their unique challenges, I would say.
Something you said then just made me think of how interesting it is that you have, coming back to the neural networks idea. So yes, humans designed machine learning, and then we remove ourselves from the process, but then we have to put ourselves back into it at the end in order to understand what it is that it's given us.
Yeah, that's such a good way to put it.
So it is an iterative process. I was also thinking how you were discussing the garbage in, garbage out, and I imagine over the years as you've been refining, working in this way, you've honed in on the best sorts of experimental design to produce the highest quality data that's going in order to make the other end of it more manageable.
That's a great point. It's not just me. We have a lot of experienced scientists on our team, the experimental team. They know the best practices and they know the data quality controls. So they do their best to design the experiments that best test the client's hypothesis. So there's a lot go on in that stage as well for our experimental platform. Yeah, definitely.
Yeah. Okay. So I want to come back to the rare diseases that you mentioned earlier, and that this is one of the ways that we're using this technology is to use worm models in order to study rare diseases. And can you give us an example, a specific example of where machine learning has aided our understanding in a rare disease or phenotype?
Yeah, we have a couple that come to mind. I think I will talk about one recent phase 1 SBIR grant that we completed is working on genetic variants in the STXBP1 gene. So this is syntaxin-1 binding protein in human, and the genetic variance in it has been associated with childhood onset epilepsy and mental delay in mental development. These are kids who might have seizure in early life and their development is delayed. That's pretty sad to see. And we have worked with clinicians that see these kids and get variants that these kids carry in the STXBP1 gene, which were thought by clinicians to be causal, very associated with their phenotype, the disease phenotype. So we have humanized the STXBP1 homologous locus in the C. elegans, meaning we're knocking the human coding sequence into that locus. On top of that, we have engineered with CRISPR point mutations that are matching the human specific genetic variants. So with that kind of human avatar model, we were then able to do a whole battery of genotyping, phenotyping assays to get a whole bunch of parameters and physiological data, and that goes into the machine learning model to try to predict the pathogenicity of the variants, whether this variant is likely associated with disease, meaning pathogenic, or whether it is actually not harmful, that is benign. So specifically, we have electrophysiological data of the pharyngeal pumping from C. elegans that is related to their feeding behavior, but it's a very rhythmic electrical activity that's generated by the muscle cell, and a lot of mutations can affect this rhythmicity of this behavior. And the SXBP1 UNC18 mutant happen to have a severe abnormal phenotype. So that's good, and we can see whether the humanization can rescue it and whether the genetic variants, that patient-specific ones, will affect this outcome. So with that electrophysiology data, we get these voltage over time or current over time measurements. So it's a time series. So that one we have to do some feature engineering based on expert knowledge. So there's two types of kind of common theme in how you get the data into the machine learning model. One route is going pure feature engineering based on expert knowledge. And the other route is more the deep learning route, which is a little bit more black boxy, but it's doing feature engineering purely with the neural networks. So we've tried both. And yeah, so the first route is we have in-house expert like our founders, Dr. Lockery and Dr. Weeks, and our senior scientist, Dr. Roberts. They were expert in the this electrical physiology in C. elegans. So with their guidance, we were able to generate some very meaningful features such as the duration, the amplitude, the frequency of the pumping behavior. And these features are kind of distilled from the raw data in a meaningful way, so they could better predict an outcome. So I use those features to model, to put in a machine learning model, and then indeed we could predict the STXBP1 mutant from the wild type. Yeah.
So is the hope then that going forward, so you must have had some set, some number of these mutations that you were querying in the model, And then now
Yes,
you've convinced
that
yourself
is
that
correct.
you've built the tool that can help you classify
Mhm.
which
Yeah, I forgot to mention that. Yeah, so we would have the training data is a set of non-pathogenic and benign variants. So this is a supervised learning framework. We have data that are labeled that we know the ground truth for. So that goes into our model. The model is trained to predict the label from the features. So because we know the ground truth, we could double check and correct our model and iteratively train it to a high accuracy in predicting these non-variants. And the hope is that with some future unknown variants, we could still use this model. It's able to generalize and tell us give us some useful hints about the pathogenicity of these unknown variants. And there are so many unknown variants.
It's just so cool to contemplate sort of how this tool is structured. Is there a minimum number of variants that have had to go into building the model to make it powerful enough so that going forward you can continue to create with new?
That's a very good question, and that's something we're still actively working on. It's because this kind of variant classification is still pretty new, and we don't really have that many variants that we have non-ground truth label for. Sometimes physicians will label it some way, and then they will submit it online to certain databases, and you will see on those databases a lot of the variants actually have mixed information. They have sort of mismatched judgment from different sources about whether they're pathogenic or not. So I say this is still pretty new, and we're trying to work on the frontier of this area.
That is so fascinating, and our timer went off just as you were closing out that thought. So Lan,
oh wow,
that was good timing. I want to thank you for taking the time again to hang out with us today and explain more about this concept of human computer interface and how that will help move forward personalized medicine. It's amazing. So stay tuned for more from us on this in the future, but I want to thank everybody for coming by and look forward to hearing from you on Facebook. So until next time, be safe, be well, take care.